Hello everyone, welcome to my channel Dr. TAPATI'S presentation. I have started discussion on biomolecules. Already I have uploaded two videos. First one was on biomolecules and introduction. Second video was on carbohydrates in detail. You should watch those two videos. In my last two videos already I have mentioned that the major complex biomolecules present in the living cells are carbohydrates, proteins, nucleic acids and lipids. In my last video I discussed about carbohydrates in detail. Today in this video I'm going to talk about proteins along with amino acids. Kindly stay with me till end of this video in order to understand most of the things associated with proteins and amino acids. I hope you will like this video. This video will be helpful not only for class 11 and NEET students but also for MBBS, IIT JEM, CUAT, GATE Life Science, CSIR NET Life Science and other BSc MSc students. So let's start the presentation on amino acids and proteins. Proteins are biopolymers composed of repeating units of amino acids. That means building blocks of proteins, those are amino acids. Amino acids are attached through peptide bonds to make polypeptide chain. Proteins are composed of mainly carbon, hydrogen, oxygen, nitrogen and sulfur elements. Enzymes, antibodies, receptors, hormones, transporters, mostly made up of proteins. Proteins are fundamental basis of structural and functional basis of life, growth and tissue repair. Amino acids. Amino acids are biomicromolecules. That means their molecular weight is less than 800 Dalton. They are organic compounds containing an amino and an acid group or carboxylic acid group as substituent on the same carbon, that is alpha carbon. This is alpha carbon and therefore they are known as the alpha amino acids. From the name amino acid, that means amino plus acid, these two groups present in amino acids. Along with these two groups, one hydrogen atom attached with the carbon, alpha carbon, and another R group which is known as a variable group or variable side chain, that is also attached with the alpha carbon. So if you overall see the structure of amino acid, you will find one amino group, one carboxylic acid group, one hydrogen atom, and fourth group is R, that is variable group or variable side chain. These four groups are attached with alpha carbon. If you want to understand the amino acid numbering, you should check this example of lysine. Lysine is an amino acid where uh, carbon numbering starts with the functional group COOH, that means carboxylic acid group, that is carbon number 1. And next carbon is carbon number 2. In this way, 3, 4, 5, 6, 6, these are the different carbon numbers where 3, 4, 5, 6, these are R group. These are present in the R group, that means in the variable group. And if you consider here which one is alpha carbon, alpha carbon is the first carbon attached to functional group. So in this case, carbon number 2 is alpha carbon and carbon number 3 is beta, 4, 5, 6, these are gamma, delta and epsilon respectively. Amino acids are substituted methane as the four substituent groups are occupying the 4 valency position of carbon. Those are hydrogen, carboxylic acid, amino acid group and variable group. In this picture also you can see, if you consider this is actually methane, so where total 4 hydrogen attached with carbon, and if you substitute one hydrogen with COOH, another hydrogen with NH2, then a third hydrogen with R group, you will get this amino acid. So amino acids are substituted methane. So but fourth hydrogen is not substituted with any group. Based on the structure of L-glyceraldehyde and D-glyceraldehyde, there are two stereoisomers of amino acids. L amino acid and D amino acid. When NH2 group present left side of alpha carbon, that is known as L amino acid, and when NH2 group present right side of the alpha carbon, and that is known as D amino acids. However, L alpha amino acids are most predominant form. L forms are used by cells because we as eukaryotes have enzymes which can only recognize L conformation. Similarly, D carbohydrates are predominant form of carbohydrates. Another fact is that four different groups are attached with alpha carbon, so it is chiral centered. Chiral centered. If you check the structure of amino acid, COOH group attached, H group attached, NH2 group attached, and NH2 group attached, and R group is attached with alpha carbon. So total four different groups attached with alpha carbon. So it is chiral center. Therefore, except glycine, where this alpha carbon is not chiral because two hydrogen atoms attached with alpha carbon. So it is not chiral center. Hence, except glycine, all other amino acids are optically active. Glycine is optically inactive because of the absence of chiral center. There are several amino acids based on nature of R group or variable group. However, those which occur in proteins are only 20 according to your NCERT book. That means mainly 20 amino acids are known as the proteinaceous amino acids because they are present in proteins. The R group in these proteinaceous amino acids could be hydrogen. If you consider about the structure of glycine, where R is equal to hydrogen in case of glycine. If you see the structure of alanine, so R group is methyl group, and in case of serine, R is equal to CH2OH, that means hydroxymethyl group. In this way, other 17 amino acids, there will be only difference in the R group. Other than that, hydrogens, amino group and carboxylic acid group, these are fixed. That means these are obviously present in each and every amino acids. Only difference would be in the R group. Again, I have written the same thing. Glycine means R is equal to H, and in alanine, R is equal to CH3 group, this one. In case of serine, R is equal to CH2OH, that means hydroxymethyl group. The chemical and physical properties of amino acids are essentially of amino, carboxyl and R functional groups. Already I told you that in every amino acid, at least one COOH and one NH2 group present. So, the properties of the amino acids are characterized by the nature of side chain group, that is R group, associated with the amino acid. If extra COOH group with R group, that is known as the acidic amino acid. If extra NH2 group attached with R, that is known as the basic amino acid. And if there is no extra NH2 or COOH group attached with R, those amino acids are known as the neutral amino acids. Let's check differences among acidic, basic and neutral amino acids. So if you consider about acidic amino acids, side chain, that means R with acidic groups or their acidic groups, in that case, extra COOH group present. Example is glutamic acid, where you will find extra COOH group attached with gamma carbon. So that carboxyl is known as gamma carboxyl group. Due to the presence of extra COOH, it is acidic amino acid. Now, if you consider about basic amino acid, in that case, side chain is with basic group, that means NH2 group. So, extra NH2 group present in basic amino acids. Example is lysine, where you will find gamma amino group. So this amino group is the part of R group or functional group, R functional group, that is the variable group. If you consider about neutral, so side chain with neutral groups or an equal number of amino and carboxyl groups. So if you see the example of valine, in that case one COOH group present and one NH2 group present. So equal number of amino and carboxyl group present. But if you consider here in the R group, there is no extra NH2 or COOH. Therefore, these are neutral amino acids. The equal number of amino and carboxyl groups, they are neutralizing each other. Therefore, these type of amino acids are neutral in nature. Already I have mentioned that according to your book, total 20 standard amino acids are incorporated during protein synthesis. So those are known as proteogenous amino acids. Among them, two amino acids are acidic amino acids, including aspartic acid and glutamic acid. Whereas there are mainly three amino acids. Those are basic amino acids, lysine, arginine, and histidine. And other 15 amino acids are coming under neutral amino acids. So in order to remember those names, you can follow this shortcut gallium p square tq CSVAG, where gallium means, in that case, G is for glycine, then alanine, leucine, isoleucine, and methionine. P square means phenylalanine and proline. TQ means threonine, tyrosine, and tryptophan. CS means cysteine and serine. Then VAG means VAG, that is V for valine, asparagine for that is A, and G for glutamine. If you don't remember this name, gallium p square TQCS VAG, at least you should remember the names of acidic and basic amino acids. It is very easy to remember because only two acidic amino acids are there, including aspartic acid and glutamic acid. And only three basic amino acids are there: lysine, arginine, and histidine. The rest of the 15 amino acids are coming under neutral amino acids. Another fact, aspartic acid, in R group, COOH group present extra, and whereas in case of neutral amino acid asparagine, instead of COOH, CONH2 group present. So that is so you can see there will be similarity between aspartic acid and asparagine structure. Only difference would be with the instead of COOH in asparagine, CONH2 group present. And similar way in glutamic acid, COOH group present in our group, but in glutamine, the similar structure is there. Only difference would be in the instead of COOH, CONH2 group present. These total 20 amino acids are encoded by 61 standard genetic code. Let's know what is genetic code shortly. Check the chapter molecular basis of inheritance in your class 12 NCERT book, and you will get to know the actual definition of genetic code. In this book, you will find this checkerboard for genetic code, and according to what is actually genetic code? Genetic code is a set of three-letter combinations of nucleotides called codons, each of which corresponds to a specific amino acid or stop signal. So according to this checkerboard, total 64 codons present containing 3 nucleotide sequences, and among 64 codons, 61 codons represent 20 standard amino acids, whereas other three codons represent stop codons. Those are UA, UGA, and WAG, that means UAA, UGA, and UAG. According to central dogma of life or central dogma of molecular biology, DNA to DNA synthesis is known as replication, DNA to mRNA synthesis is transcription, and mRNA to protein synthesis, that is known as translation process. So during translation process, based on the sequence present in mRNA, there will be sequence of amino acids in protein. Suppose this is 7 codons present in mRNA. So depending upon the seven codons, during the protein synthesis, amino acid sequence will be total 7. Methionine, phenylalanine, phenylalanine, phenylalanine. So total 6 phenylalanine with 1 methionine. So this is the actual sequence in the protein based on codons present in mRNA. DNA is composed of ATGC nucleotides, whereas mRNA is composed of AUGC nucleotides. Three nucleotides together representing one codon, and based on the codon sequence present in mRNA, there will be formation of protein and based on codon sequence, amino acids are attached through peptide bonds. So suppose codon 1 is representing amino acid 1, so second sequence of amino acid will be based on codon 2 present in mRNA. Although I told you about 20 proteinaceous amino acids, but selenocysteine and pyrrolysine, these are 21st and 22nd amino acids, and they are also inserted into proteins, and they are mostly encoded by stop codons. Like selenocysteine is encoded by UGA, which is stop codon. Similarly, pyrrolysine is encoded by UAG, and that is also another stop codon. Selenocysteine has similarity with cysteine. In cysteine, sulfur present, whereas in selenocysteine, at that place, selenium present. And similarly, pyrrolysine has similarity with lysine, where NH2 group present in lysine, but in case of pyrrolysine, pyrroline ring present. Just few minutes back, I told you amino acids are classified into 3 groups: acidic, basic, and neutral. Although above 500 amino acids exist in nature, but overall total 22 genetically encoded or proteinogenic amino acids are there, where 20 amino acids are encoded by standard genetic codes, whereas additional 2 amino acids known as selenocysteine and pyrrolysine, those are encoded by stop codons UGA and UAG respectively, and they are also incorporated during the protein synthesis or translation. However, based on polarity, amino acids can be classified into 2 groups: hydrophilic or polar, and hydrophobic or non-polar amino acids. Hydrophilic or polar amino acids are of 3 types: positively charged, negatively charged, and uncharged. Positively charged amino acids examples are lysine, arginine, and histidine. They have extra NH2 group in R group, whereas in negatively charged amino acids including aspartic acid, glutamic acid, they have extra COOH group in R. And uncharged, if you consider, examples are serine, threonine, tyrosine, and cysteine, where serine, threonine, and tyrosine, they have extra OH group in R, and in cysteine, extra SH group present. So uncharged means they are uncharged, but due to the presence of OH group and SH group in R group of amino acid, they are acting as the hydrophilic or polar amino acids. And another fact is that tyrosine is aromatic amino acid, but still it has polarity due to the presence of OH group. Now, if you consider about hydrophobic or non-polar amino acids, you can categorize into 2 groups: aliphatic and aromatic. Aliphatic amino acids are glycine, alanine, valine, leucine, isoleucine, proline, and methionine. Aromatic amino acids are tryptophan and phenylalanine. Aromatic amino acid, I told you tyrosine is also aromatic, but it is uncharged and polar amino acids. But tryptophan, phenylalanine, those are non-polar, that means hydrophobic amino acids. Now, if you consider methionine, this is considered as an aliphatic amino acid, even though the side chain contains a sulfur atom like cysteine, but it is fairly non-reactive like the true aliphatic amino acids. Therefore, it is coming under aliphatic amino acid or non-polar amino acid here. A particular property of amino acid is the ionizable nature of NH2 and COOH groups. Therefore, the structure of amino acids changes in the solutions of different pH. That means charge of amino acid varies with pH of the media where it is dissolved. Need charge of amino acid will be 0 when pH value is equal to pi. So pi means isoelectric point. So isoelectric point is the pH at which need charge of the amino acid is 0. When need charge of amino acid is 0, at the condition where COOH group present in deprotonated form as well as NH2 group present in the protonated form. So positive and negative, one positive charge, another negative charge. So total need charge is 0 in this case, and this form is known as the zwitterion form. So at pi, amino acid present in zwitterion form where need charge is 0. Whereas if you increase the pH of the solution, and if pH is higher than pi value of the amino acid, at that condition that amino acid will be in the negatively charged, because of the fact that at that pH COOH group is present in deprotonated form, but NH2 group remains as it is. Similar way, when pH value is less than pi, at that condition, that amino acid will be present in the positively charged condition because of the fact that NH2 group is in protonated form, but COOH group is not in deprotonated form. So positive charge is present. So this amino acid is positively charged. So what I told you, pi means the pH at which need charge of an amino acid is 0. So that is known as the isoelectric point or pi. When pH of the solution is greater than pi value of the amino acid, at that condition amino acid carries a net negative charge. Whereas when pH value is less than pi value, at that condition amino acid carries positive charge, and when pH value is equal to pi value, at that condition amino acid need charge is 0. Nutritionally, amino acids are classified into main two groups: essential amino acids and non-essential amino acids. Essential amino acids can't be synthesized by our body, hence must be supplied from outside through diet or food, whereas non-essential amino acids can be synthesized by our body even if we do not get them from diet or food. There are mainly 9 amino acids. Those are known as essential amino acids. You can shortcut way remember it as a HLL private team, HLL PVT team. And for the non-essential amino acids, remember gas PCT. And another category is also there, that is known as semi-essential amino acids. So in that case, less they are synthesized less in infant, children, and pregnant women during lactation also. Examples are histidine and arginine. Let's start discussion on proteins. Proteins are complex biomolecules. Those are biomacromolecules with molecular weight greater than 800 Dalton. Proteins are polypeptides, linear chains of amino acids linked by peptide bonds. A protein is heteropolymer of 20 or 22 types of amino acids. These are not homopolymers because a homopolymer has only one type of monomer repeating a number of times. In this case, in protein, more than one type of monomer present. So proteins building blocks are amino acids. Proteins carry out many functions in living organisms. Proteins are fundamental basis of structural and functional basis of life, growth and tissue repair. Some proteins fight against infectious organisms, as for example antibodies, which are made up of glycoproteins. Some proteins are hormones like insulin, enzymes like pepsin, trypsin, receptors like receptors for hormones, smell and taste, and pigments, as for example hemoglobin. Some proteins transport nutrients across the cell membrane, as for example, GLUT4 for glucose transport. Most abundant protein in animal world, that is collagen, whereas most abundant protein in biosphere, that is rubisco. Rubisco means ribulose bisphosphate carboxylase oxygenase, which is required for the photosynthesis. These are the elemental composition of proteins. Carbon 50 to 55%, hydrogen 6 to 7.5%, oxygen 19 to 24%, nitrogen 13 to 19%, sulfur 0 to 4%, and other elements like different metal ions and non-metals. Nitrogen is an essential component of proteins. The amount of protein in biological fluids and foods can be measured by estimating nitrogen content, mostly by following the method, Zeldal method. Already I told you, in a polypeptide or a protein, amino acids are linked by peptide bond, that is CONH bond. It is found when the carboxyl, that means COOH group of one amino acid reacts with NH2 group of next amino acid with the elimination of water. That process is known as the dehydration due to the elimination of water moiety here. If you check the characteristic of peptide bond, those are partial bond, partial double bond characters. Due to that, it is planar and rigid, so no freedom of rotation. And the C-N bond is in trans configuration. Biologists describe the protein structure at 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. If you consider about primary structure, the sequence of amino acids, that means the positional information in our protein, which is the first amino acid, which one is the second amino acid, and so on, is called the primary structure. And a protein is imagined as a line. The left end represents the first amino acid, and the right end represents by the last amino acid, and the first amino acid is called as N terminal N, and last amino acid showing you the C terminal end. This is a simple primary structure of a protein containing 7 amino acids, and those are attached through 6 peptide bonds. So when 7 amino acids present, total 6 peptide bonds present here. But if you consider when 10 amino acids present in a protein, in that case 9 peptide bonds possible in between those 10 amino acids, because in between 2 amino acids only 1 peptide bond possible. And extreme left part of the chain is known as the N terminal end, because NH2 group is free here, whereas right corner of the chain is known as the C terminal, because COOH group of last amino acid is is free. COOH group is free in that case. Secondary structure means folding and twisting of polypeptide, that means primary structure of protein, due to the hydrogen bonding between hydrogen of NH and oxygen of C11O. And in that case, in case of alpha helix secondary structure, intramolecular hydrogen bonding present, whereas in beta secondary structure, beta sheet, in that case intermolecular hydrogen bonding present. So alpha helix is like staircase, revolving staircase, whereas if you consider about the beta sheet, so it is it is like zigzag pattern, where intermolecular hydrogen bonds present between two beta sheets. Now, if you consider the examples of few proteins containing secondary structure, in that case, myoglobin contains alpha helical structure, collagen contains triple helix structure, keratin, alpha and beta both structures present. In silk fibrins, beta sheet present. two figures are given. First one is secondary structure of protein, and this is alpha helical structure, and it is like revolving staircase. Whereas if you consider about the tertiary structure of protein, so the second figure is showing you the tertiary structure of protein, where the long protein chain is folded upon itself like a hollow woolen ball, giving rise to this tertiary structure, which is three-dimensional view of protein. And this tertiary structure is absolutely necessary for the many biological activities of protein. So in order to give the in order to show the activity of protein, tertiary structure is most of the cases required. Whereas if you consider about the quaternary structure of protein, some proteins are an assembly of more than one polypeptide or subunits, and the manner in which these individual polypeptides folded polypeptides or subunits are arranged with respect to each other. That means either a linear string of sphere arranged upon each other in the form of a cube or plate, etc., is the architecture of a protein, otherwise known as the quaternary structure of a protein. So quaternary structure is observed mostly when any protein consists of more than one polypeptide subunits, and in that case those several subunits, how they are arranged together, how they are bonded together, folded together, and after folding they are giving quaternary structure. And in that case quaternary structure is showing you the most active form of the protein. So with the quaternary structure they will show you biological activity when proteins are with more than one polypeptide subunits. As for example, adult human hemoglobin consists of four subunits, two alpha, two alpha and two beta subunits present in hemoglobin molecule. So two of these are identical to each other. So two alpha, hence two subunits are two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin, and that is giving the quaternary structure of hemoglobin. In a shortcut way, if I explain primary, secondary, tertiary, and quaternary structure, I can say that primary structure means the sequence of amino acids. That means amino acids attached through peptide bonds, where left end is known as the N-terminal end and right end is known as the C-terminal end. Secondary structure means folding and twisting of that primary structure. So it may be alpha helical structure or it may be beta sheet structure. Whereas if you consider tertiary structure means folding of the secondary structure, giving you woolen ball-like structure, and that is known as the tertiary structure. Whereas if you consider about quaternary structure means when any protein contains more than one subunit, so those subunits, folding of those subunits uh, can give you quaternary structure. This table is showing you different forces stabilizing structures of proteins. In case of primary structure, peptide bonds present. In case of secondary structure, peptide bonds and hydrogen bonds present. In alpha helical structure, intramolecular hydrogen bond present, whereas in beta sheet structure, intermolecular hydrogen bonds present. Now, if you consider about the tertiary quaternary structure, in that case, covalent bonds as well as non-covalent bonds are there. Covalent bonds means peptide bonds and disulfide bonds present. And in case of tertiary structure, intramolecular disulfide bond present, whereas in intermolecular disulfide bonds present in quaternary structure of protein. Non-covalent bonds are hydrogen bonds, hydrophilic interaction, hydrophobic interaction, ionic interaction, van der Waals interaction, and salt bridge interaction. Salt bridge means ionic bond plus hydrogen bond together. This figure is also showing you forces stabilizing structure of protein. Along with the polypeptide backbone, ionic bonds are there. So ionic bond possible in between positively charged amino acid and negatively charged amino acids. So here aspartic acid is negatively charged amino acid and lysine is positively charged amino acid. So in between these two amino acids, ionic bond possible. Hydrophobic interaction possible in between aliphatic amino acids. As for example, valine and alanine both are aliphatic amino acids. So they are showing a hydrophobic interaction. Then a disulfide bond present in between cysteine residues, and this is intramolecular disulfide bond within a single polypeptide chain. After that, fourth interaction is hydrogen bond. So hydrogen bond is possible in between hydrogen and any negatively charged element. So here serine and asparagine both are in between these two amino acids, hydrogen bond possible. Then fifth interaction is hydrophilic interaction. So it is possible by hydrophilic amino acids. As for example, serine is hydrophilic because of the presence of OH group. And sixth interaction is van der Waals interaction. So van der Waals interactions occur when adjacent atoms come close enough that their outer electron cloud just barely touch. So these are overall to, six interactions, and seventh interaction is salt bridge interaction. Salt bridge interaction means ionic bond as well as hydrogen bond. So in between aspartic acid and lysine, ionic bond is present as well as hydrogen bond is present together. So it is showing you the salt bridge interaction. So total 7 interactions possible in tertiary structure, and similarly in quaternary structure also. Total 7 types of interaction possible. If you think this video is helpful for you, kindly like, share and subscribe this channel.